Good morning and good afternoon. My name is Carl Latham and I'm Vice President of Sales at Ontoforce, and I'm delighted to introduce this webinar on data-driven efficiencies in clinical studies supported by the DISQOVER knowledge platform alongside our partners Clarivate. Joining me on the call today, we have Tamara Stankovic, Customer Data Scientist at Ontoforce, Dr. Ketan Patel, Vice President of Qualitative Product Platform at Clarivate, and Dr. Peter Revel, Product Manager at Clarivate. Before we dive into today's case study demonstration, I will share a brief introduction on Ontoforce before handing over to Ketan. So Ontoforce is a life sciences knowledge platform company, and we help customers to unlock hidden insights from data by breaking down boundaries between silos to make data discoverable, accessible, and usable. Oh, sorry. Sorry, my mouse is going a little bit crazy this morning. So yes, it's a well-established fact that data is growing exponentially in complexity. And our clients share with us that they face a multitude of challenges when looking to understand how to leverage the power of the information that they can access. At a user level, the problem becomes more complex. So researchers, scientists, and business users will have many questions or hypotheses that they want to test every day. So you could be looking for knowledge about a specific patient population, information to support clinical study design, or a competitive intelligence question. And the issue that the user faces is that they are searching information across different data sets that are heavily siloed. And furthermore, it's often the case that there might be resources available that the user doesn't even know about. It's really time consuming to go to all of those resources, and data is not consolidated in a consistent, harmonized, or searchable way. So for example, one gene database might refer to EERP when another refers to EGFR. So how can Ontoforce help with that? Well, we can integrate complex, unstructured, and siloed data from internal, public, and third-party sources into our DISQOVER platform. And through our integrated knowledge graph, users can access unified information very easily, and the knowledge graph can be applied to multiple use cases. DISQOVER itself is a self-service data discovery platform for business users, meaning that everyone should be able to find and access knowledge. And using DISQOVER, you can customize the outputs of your results to create a very intuitive user experience specific to your use case. And DISQOVER can also embed alongside your existing IT ecosystem with custom applications like Chemaxon, Spotfire, and 7bridges to further optimize the user experience. Our engagement model is to work with clients on defined use cases that span the entirety of the drug development lifecycle. And here we can create outputs linked to defined outcomes with a clear, quantifiable return on investment for our clients. And should you be interested in learning any more after this demonstration today, my details will be shared at the end of this webinar. Thank you, and I'll now hand over to Ketan.
Thanks, Carl. So if you go to the next slide, please. Yeah, this is just an introduction. Hi everyone, my name is Ketan. I'm the Vice President of the qualitative Product Platform, which is our flagship life sciences platform at Clarivate. And I'll talk a little bit about that in a moment. So Clarivate actually helps our customers from very early stages of research and drug discovery all the way through to clinical development, regulatory affairs, and market access. For this demonstration, for this webinar, we're going to focus on the clinical trials aspect of it. In this case, Clarivate provides data on protein-protein interaction networks, pathways, genetic biomarkers, drug discovery, and also clinical trials. And we also have a large consulting team that helps our customers develop use cases in these areas, as well as utilizing our data. But one of the things that a lot of our customers really like about our data is that it's integrated and it's all manually curated as well. So, you have the public domain data, but then you have also manually curated data, which is of high quality and indexed with specific ontologies and controlled vocabularies. And that's where Clarivate brings a lot of data to the table. We also then provide this data in a number of ways to our customers, including shrink wrap products, but also through our APIs and data feeds. So if you go to the next slide, in our collaboration with Ontoforce, we've leveraged our existing Clarivate APIs to be able to integrate with the Ontoforce DISQOVER platform. And for this demonstration, you'll be seeing a number of different data sets, including our investigational drugs data set, which includes over 80,000 drug program information. Also our clinical trials intelligence data, which includes over 500,000 clinical trials, and also our biomarkers database, which includes about 48,000 biomarkers. These are the data sets that we've integrated, and over time, we will be collaborating with Ontoforce to bring many more of our APIs and data sets to the DISQOVER platform. And so you'll be seeing how these integrated data sets can be utilized alongside publicly available data to solve real problems in clinical research. And I'm now going to hand it over to Peter.
Thanks, Ketan. So to demonstrate the breaking down the silos that's possible with DISQOVER, we're going to use the story of KRAS gene and focusing in on lung cancer. First of all, to introduce myself, Peter Revel. I'm a product manager with Clarivate and with experience in content management before that, and in biotech startup and academia. Joining me as well for the demonstration is Tamara from Ontoforce. So over to you, Tamara. Okay, shall I continue? Just nod. We've lost the presentation there.
Okay, so we had a bit of a technical issues, but it is resolved now. Hi, my name is Tamara, and I work as a data scientist at Ontoforce. When I started working as a junior scientist at University of Belgrade, and then continue my journey as a bioinformatics analyst at Seven Bridges. And yeah, I will help Peter to do a demo on DISQOVER platform today.
Thank you. If we could have the slides back, then we'll move to the next slide. So today in the demo, I'll give a couple of slides of introduction to the topic as refreshers on the biology of KRAS-driven disease, and then we'll move into the demo itself. So just as this, if you could click into the display view here. And we'll use DISQOVER to really look at two aspects of KRAS-driven cancers. One is around the early discovery, particularly the target validation, looking at gene variant disease associations and target validation. And then we'll move through to the clinical, and particularly they're looking at KRAS as a biomarker for patient selection. Okay, on to the next slide. So into the biology first, two background slides. This one illustrating the point that lung cancer is multiple diseases, whether you slice it by histology to see different disease types there, or by biomarker, again defining the disease by the oncogene here. And you can see in this biomarker panel, the pie chart here, well-known gene variants driving the disease, some of which are druggable. For example, the EGFR, the ALK, the BRAF, and HER2, all with drugs targeting those. But a big unknown factor in this, at least for the last 30 years, has been the KRAS, which has been particularly slippery in terms of drug development as a target. I will go into that in a second. So moving on to the next slide, here getting into the granular details of KRAS-driven lung cancer in this case. So you can see across the top, just below the membrane there, is KRAS, which is in two forms in equilibrium, the on and the off form, driven by this exchange of GDP-GTP. GTP is the activated, bound form is the activated form. And in the mutant version, the glycine amino acid number 12 to cysteine substitution, this favors the equilibrium towards the on form. So KRAS is on through the mutation. When it's on constitutively, it is driving multiple signal pathways through RAF, MEK, ERK, through PI3K, mTOR, through the RAL, PD-L1 mechanisms, all of those leading through to cell proliferation, tumorigenesis, and the cancer. In this example that we're showing, lung cancer. You can see here at the very top, two drugs in development, one of which is just recently approved, is sotorasib, that's specific for the KRAS glycine 12 cysteine mutation. And we'll be taking a look at that in the demo as well. So that's the background for the demo today. Before we do the demo, I just want to go back one slide, the topics that we'll cover. If we go back one slide here, and we will. Okay, so the 3 points that we'll cover in the demo are: one, we'll take a look at the strength of evidence for the gene variant disease association. Secondly, we'll take a look at KRAS as a druggable target. And then thirdly, we'll move into the clinical trial strategy using KRAS as a biomarker for patient selection. Okay, so Tamara, if you could display the screen there, and we'll go into the demo. Okay, so here's the starting panel, if you like, with multiple content sets in here. So this first start, we're starting from KRAS. And you'll see here as we move through this very quickly through to the gene, from there filtering down to the human specific gene, and from there to the variants. And you see as we come into the variants here of the human KRAS gene, over 24,000 variants here, of varying significance for the clinical validity. Up at the top here, as Tamara was just showing there, the various data sources here. So this is all public data coming, being pulled into the dashboard here for these 24,000. And now moving down, coming through to the clinical information. So we're looking for biomarkers for disease for non-small cell lung cancer. So allowing us to narrow down to a subset here, and particularly clicking further into the pathogenic based on clinical significance. So you can see there with just a very few clicks coming down to 25 variants of pathogenic significance for non-small cell lung cancer. This and in there you can see the RS number ending in number 530, which is the encodes at least one of the alleles in there encoding the glycine to 12 cysteine substitution in codon 12. If we look, so you can see again the data sources there, and if we scroll down, although we've filtered for non-small cell lung cancer, you can see a range of other diseases here. So this set of variants here are biomarkers for multiple diseases, which is important because if you take a strategy to target any particular variant or any particular gene, it's not just the disease you're aiming for, but it's the white space of other conditions that are treatable as well through targeting this gene. So this is the first bit that we wanted to show, which is fairly quickly zooming in on a subset of variants that are pathogenic for the disease. Next is we're taking a look at the target druggability. So for over 30 years, been various attempts to drug this target. And you can see here what Tamara has done is looked at KRAS. Actually, I just want to scoot up a little bit and look at some of the other information before we go into the mechanism.
Okay.
So here, yes, take us to the mechanism, then we'll take a look at the dashboard. Yeah.
Yep.
So fairly quickly, using mechanism, coming down into a subset of drugs based on KRAS inhibitors. And you can see over on the left-hand panel at the top is that drug we've seen before, sotorasib, which is already approved, recently approved for this. I just want to take a little look at the rest of this panel here. So you can see the various phases of development, a fairly strong pipeline of other drugs coming through. There is information about the company originators, so allowing you competitive intelligence if you wanted to. There are other dashboards here as well, for example, the patent dashboards to understand freedom to operate. But today we're going to move through from where we are now, which is this set of 72 drugs, and we'll look at these trials associated with these 72 drugs. So we've used the DISQOVER platform with the public data to look at gene variant disease association, establish a subset of gene variants that are pathogenic. We've used the DISQOVER platform with Clarivate data to demonstrate that the target is druggable. And here, now we've moved into the clinical trial data, again from Clarivate, and we're looking at these drugs and the 34 trials, at least in this data set, that are related to those drugs. I do want to take a moment just to look through this dashboard before we filter it down further. So for these clinical trials, again, you can see under indication the various trials that are underway for the different indications, and the drugs being used for those indications. The phases, again, showing a good, healthy pipeline and a distribution through those trials. This biomarker panel, biomarker name panel, is, as you can see here, as Tamara clicked on that, we can see 34 clinical trials using the KRAS as a biomarker. So every single one of these trials, all using KRAS, and that makes sense because it is the druggable target here. Other biomarkers here of interest, so there's STK11. I think I also saw PDL1 in there, and I know that these trials are also using tumor mutation burden as well as a biomarker. So you can see that the development programs here and the trials, those developing these KRAS targeting drugs, are also using biomarkers that predict efficacy for immune inhibitors. Or the immune response drugs here. So if I were looking at the data based on those trials, I would be looking to understand what's the relationship, for example, between these immune predictive markers and the KRAS inhibitor response in these trials. Coming down here, so patient segment is a specific way of filtering the data set down. And what's beautiful of this filtering here is that we can go really specific and come down to the particular variants, to the variant level here. So what's being shown here in this part of the workflow, selecting a few of the single nucleotide polymorphisms that are associated with the allelic change that leads to these set of variants that drive the disease. And so we've narrowed the numbers of trials down here to 14. And let's take a look at the story, particularly for sotorasib, which is the approved drug. And you can see here we've got down to 3 trials for sotorasib. And this top one here, this phase 1/2 study, we move through, and again, you can see the supporting information for this trial. And in particular, I want to go through to the press release data there. So this is all supporting information linked to that trial. If you search by FDA, we're filtering the press releases down now here, and you can see the second one there is the statement from the FDA, the press release from the FDA, where they approved this drug. So what this is telling us is that based on data from this phase 1, phase 2 clinical study, the FDA approved this drug for use in lung adenocarcinomas in patients who had previously been resistant to other therapies. So I just want to draw a little bit of a summary here of where we've gone as we've walked through the DISQOVER platform. We started broad with lung cancer, stepped down to KRAS, did the search with KRAS, found 34,000 gene variants, and very quickly, using public data, came down to a set of 25 variants that are clinically significant for lung cancer. Then we asked to what extent is KRAS druggable, and we used the Cortellis data to, in this platform to show a pipeline of drugs coming through which are targeting KRAS, and the first of these approved, as you can see here, in May of this year. So yes, it is druggable, and particularly in relation to the gene variant. Then we stepped in again to the clinical trial data, and we're using the patient segment. We were able to fairly quickly again narrow down the subset of trials, and this is where this biomarker-driven approach has really supported the drug development, is using the variant as a mechanism to select patients, to enrich the trial for patients who are predicted to respond to the therapy, which allowed the drug sponsors to very quickly demonstrate efficacy to the point that the FDA was prepared to approve this drug on the basis of this phase 1/2 study. So a really strong biomarker approach in the clinical data trial here to drive the drug development. So that draws to a close the demonstration portion of this presentation, and I'd like to hand it over again for questions.
Thank you for that, Peter. So yes, if you've got questions today, can I please ask you to pop those questions into the chat, and I will share those with Peter and Tamara for comment. I'll give that a few seconds. Okay, so we've got one question here, Peter, which was during the demonstration today, is this using all of Cortellis's available data, or is it specific subsets of data?
Okay, so there's 2 answers there. So this was a specific subset that was loaded for the demo purposes. So for example, you saw we stepped down from 72 drugs to 34 clinical trials. That's not the full picture of KRAS and the drugs, the drugs targeting. That's just the picture of the data that we loaded, the demo set. So somebody using the full Cortellis API would see the complete picture there.
Okay.
The other answer to that question is we showed data from the drug development API and the clinical trial API. And you could see as well in there how the two connected fairly smoothly in that flow, hence breaking down the barriers. But there are other APIs that are relevant to this story, for example, the biomarker API. And again, that would allow the smooth transition, for example, to go from the clinical trials we saw to expand out again and see the other biomarkers that we use in that clinical trial and start to explore there.
Thank you. Another question we've got is, this might be a question for Ketan and Peter. How easy is it to set up the API licenses for the data sets that we saw today?
Yeah, I can answer that, Carl. So basically, if you are a customer of the Cortellis APIs already, then and you decide to be a customer of Vontaforce, then you can turn this functionality on like today. But if you don't have access to the Clarivate APIs, then it's just a simple case of licensing the relevant data sets and APIs, and we can help with that. Take you through the different APIs that we have, and based on your use cases and based on what you want to do, we can recommend which APIs to license, and then sort you out with a trial and yeah, get started. I would just add that we are at Clarivate working on more advanced APIs where we can slice and dice the data even further. So look out for these next year, where we'll be able to kind of cut the data even finer. So if you have a small use case, then you can license a subset of our data, and that will be made available as an API. So yeah, but we can work with you on that.
Great, thank you. Got another question that's come through on the chat. How complicated is it to integrate additional data sources, e.g. Safety information coming from OffX API into the analytics platform? Is it just plug and play by ticking a box? It might be one for Tamara.
Yeah, so for us, it would be just plug and play. So as Ketan mentioned, like all of these different APIs from Cortellis, like we use them as bricks. And then once a customer actually has a license for a specific API, for us, it's just really easy to build and to integrate with all the different public data sources that we can provide with DISQOVER.
Yeah, and is it also fair to say it's the same for if can we integrate information as easily if it's not in an API format?
Yes, that is. Yes, so that is also something that cannot be done. So it's not always the case that we have data sources with as nice API structure as Cortellis has. So even if you have something in CSV formats, in Excel spreadsheets, like internal data, that is something that can be easily integrated and incorporated in DISQOVER.
Thank you. Let's see any more questions on the chat. Do we have any final questions? yeah, less of a question, but just a good bit of feedback from the panelists that it was a nice example of linked data enabled biomarker approach to drive drug development. So thank you for that feedback. We do have one other question. Is Web of Science data also part of the API offering? One for our friends at Clarivate.
Tamara, do you want to answer that? I mean, there is a WAS API.
Yeah. Yes.
I believe that it's possible to integrate in, but I'm not sure if we've tried that yet.
Yes. So we have already started on integrating WAS. So there is a pipeline ready and a very small subset of WAS currently in DISQOVER. Yeah, so it is not something that we were focused on today, but yeah, it is something that we have already tried and that is integrated.
We have another question coming through. I think one for Clarivate. How is patient profile handled in Clarivate, and could it be useful for OntoForce?
Not sure what is meant by patient profile. We do at Clarivate have additional data sets which relate to kind of patient level data from real world sources. And that's something that some of our customers have looked at integrating into things like OntoForce. But it's a separate data set from the Cortellis APIs. And it's a lot more complex because because it's patient level data, there are certain use case rights and restrictions, etc., that we need to take into account. However, the data, the patient profiles are all de-identified, and depending on the use case, it could be integrated.
Thank you. Got a couple more questions coming through. So when we were looking at the canonical types at the very beginning of the demonstration, where are the literature hits coming from? Are they full text or abstracts? And are these typically unstructured or structured content sets?
I could take that one. So I can certainly speak to the Clarivate process. So this is full text literature. We do have teams of scientists who read the full text and are trained to index using the controlled vocabularies that we've demonstrated here today. So for example, the patient segment in a clinical trial, and the multiple sources as well. We saw press release, but also taking data from clinical trial registries, from published results in journals, from conferences, so it's giving a really timely response there. And then not relevant for clinical trials, but for some of the other data sets as well, data taken from patents. And so a good range of source documents there, and all indexed by scientists using full text.
Thank you for that. And then another question, what's the Clarivate strategy to have more APIs linked to DISQOVER? And is this only for Clarivate internal use or for all Clarivate customers?
So I can answer the second part, which is, yeah, it's for joint Clarivate and OntoForce customers, of course. And I can also confirm that we already have a couple of customers who have used Clarivate data via the OntoForce DISQOVER platform and integrated it with their own data sets and into other data from other providers as well. In terms of what's the Clarivate strategy to have more APIs linked to DISQOVER, our aim is to have all the APIs linked to DISQOVER eventually. But we've started off with the kind of more popular use cases around clinical. I think the next stage will be moving more into discovery with the preclinical drug discovery data, as well as the systems biology data for our colleagues in discovery and preclinical. And then the final set of APIs would be more downstream in terms of regulatory data, regulatory approval packages, as well as HTA submissions data, which we also have linked to clinical trials. And that's also very important for, or useful for evidence generation and medical affairs and health economics and outcomes research.
Great, thank you, Ketan. I think that's that in terms of questions. So yeah, maybe we can just go to the wrap, last slide. Yeah, so if you've, what I'd like to share is that if you've seen through going through the examples today, if you have any further questions about the use case that we presented today, or any other questions around the application of the DISQOVER platform, or you'd like to know more about the Clarivate APIs, one way of getting in contact with us is through this email channel, which is sales@ontoforce.com. And then on the next slide, there's also a link to contacting Clarivate directly. However, we will share in the post-webinar communication ways of getting in contact with representatives at both Clarivate and OntoForce. So I think that just leads me to say thank you very much to Tamara, Peter and Ketan for supporting this webinar today. I hope you've obviously found it a valuable use of your time. We would also welcome any feedback in terms of other areas that you would like to see Clarivate and OntoForce cover in terms of future webinars. We'd be very happy to receive that feedback from you. But yeah, I'll close on saying thank you very much, and I wish you a pleasant morning and afternoon wherever you are in the world watching this.
Thank you. Bye.
Thank you.
Thank you.